Hello everyone, I hope you all are fine. So today our topic is Meselson and Stahl experiment which proved semi-conservative mode of DNA replication. So as you know, there are three theories on DNA replication. Yes, one is conservative, one is semi-conservative, one is dispersive. So Matthew Meselson and Franklin Stahl experiment, which was performed in 1958, proved the semi-conservative mode of DNA replication. So in semi-conservative mode of DNA replication, always two daughter DNA molecules are produced from the parent DNA molecule, and each daughter molecule consists of one old strand, that means one parent strand, and one newly synthesized strand. Okay, this is called semi-conservative mode of DNA replication, and this mode of replication was proved by the experiment which was performed by Meselson and Stahl. Okay, so for this experiment they actually experimented on E. coli bacteria to prove this supposed mode of replication. They experimented on E. coli bacteria. Remember, they experimented on E. coli bacteria. And always remember, the E. coli bacteria divides in 20 minutes. That means the cell division occurs at every 20 minutes. Okay. So now say they conducted an experiment on E. coli bacteria which divides in 20 minutes. Okay. Then for this experiment, Meselson and Stahl used two radioisotopes of nitrogen. Remember, for this experiment, Meselson and Stahl used two radioisotopes of nitrogen. One is the 15N, one is 15N, which is called as the heavy isotope, and one is 14N, which is the light isotope. Okay. So now these two heavy and light, these two isotopes, one is the 14N, one is 15N. These two isotopes of nitrogen can be distinguished based on their densities by centrifugation in cesium chloride. So what is cesium chloride? So cesium chloride is a highly water soluble heavy salt. Okay. So Meselson and Stahl proved the semi-conservative mode of DNA replication, and they experimented on E. coli bacteria which divides in 20 minutes. Okay, and for this experiment they used two radioisotopes of nitrogen. One is the 15N, which is called as the heavy radioisotope. One is the 14N, which is called as the light radioisotope. And this heavy and light radioisotope of nitrogen can be distinguished, on their densities, on the basis of their densities by centrifugation in cesium chloride. Remember, when these are centrifuged in the medium called cesium chloride, then we can differ, or we can distinguish the heavy one and the light one. Remember, the medium where these are centrifuged is the cesium chloride. Okay, and what is cesium chloride? Cesium chloride is a water soluble salt, heavy salt. Okay, so now let's move to our next slide. Here the steps of the experiment. So now they cultured, for this experiment they cultured the E. coli bacteria in the medium containing the heavy isotope of nitrogen 15. Okay, always this is the ammonium chloride. This compound is called ammonium chloride, where the nitrogen is labeled by the heavy isotope 15N. Remember. So for this experiment, Meselson and Stahl cultured E. coli bacteria. These are the normal fresh E. coli bacteria in the medium, in the nutrition medium containing the heavy isotope of nitrogen 15N. The heavy isotope is 15 and the light isotope is 14. Okay, so over many generations they cultured E. coli in this medium over many generations until 15N isotope was integrated into bacterial DNA. Okay, That means they prepared one medium and mixed the radioisotope 15N in that medium, and they grew the E. coli bacteria in that medium which contained the 15N isotope, which is the heavy isotope, and they grew this E. coli bacteria in that medium over many generations until 15N was completely integrated into that bacterial DNA. So now you can say that E. coli DNA only contain the 15N isotope. Okay, remember this point. After that, Meselson and Stahl transferred this E. coli bacteria. Okay, transferred this E. coli bacteria from the 15N medium to a new medium which contain the light isotope of nitrogen, which is 14N. Remember, this is the compound where the nitrogen is labeled by the radioisotope. Okay, that means when they grew the E. coli bacteria in the medium containing 15N, okay, in the medium 15N, then they transfer all this E. coli bacteria from this 15N medium to a new medium which is the 14N medium. That means now the second medium contain the light isotope of nitrogen, which is the 14N. So I will show you in the diagram later. Okay, first remember the steps. First, they prepared the 15N medium. Now they transfer this bacteria from 15N to a new medium where they added the 14N radioisotope. Okay, so after first generation, that means after first cell division, when the E. coli bacterial cell divides, then along with the cell, the DNA also divides. Okay, so now after first generation in the medium containing 14N, so after first generation, the DNA replication occurs in the 14N medium. Okay, when the cell divides in 20 minutes, in first 20 minutes after transferring into the new medium, then here see, after first generation, the E. coli divides in 20 minutes, remember, in the medium containing 14N. Okay, now the DNA was tested for the heavy isotope of nitrogen through the density gradient centrifugation using cesium chloride. So when they transferred the bacteria from the 15N medium to 14N medium, after 20 minutes the bacterial cell divides, and along with the cell division the DNA also divides. That means the two new DNA molecules are formed from the parent DNA, which is now known as the first generation. So after first generation, they tested the DNA of the first generation for the heavy isotope of nitrogen through density gradient centrifugation using cesium chloride. Okay, so after that, when the DNA is mixed with cesium chloride, remember, when the DNA is mixed with cesium chloride, then it will settle down at a particular height in the centrifugation. Remember, heavier towards the base and lighter one higher up. That means simply, when the labeled DNA molecule is mixed with the cesium chloride, which is the heavy salt, remember, when the DNA which is already labeled with radioisotope is mixed with cesium chloride, then it means what? It means the DNA which is labeled by the radioisotope, it will settle down at a particular height in centrifugation. That means after centrifugation in that container, this DNA will settle down at a particular height. Okay, the heavier one towards the base and lighter one higher up. That means always that DNA which is labeled by heavy isotope. Okay, the DNA which is labeled by heavy isotope, in this case the heavier one is the 15N and lighter one is the 14N. So the heavier one settle down towards the base and the lighter one higher up. Okay, do you get my point? That means always the DNA with labeled with, that means the DNA with the higher or the heavier isotope settle down at the bottom part, and always the DNA labeled with the light isotope remain in the upper part of that container after the centrifugation process. Okay, now let's move to our next slide. So Meselson and Stahl found that the DNA of the first generation was hybrid or intermediate. That means when they observed that two new DNA molecules after first generation, they found that each DNA molecule contain both 15N and 14N strand. That means these two new DNA molecules are called as the hybrid or intermediate. Okay, do you get my point? That means after first generation, when Meselson and Stahl observed the new generation, then they found that the product of first generation were hybrid or intermediate. That means the both strand contain 15N and 14N strand. Okay, clear? Again, after 40 minutes, 40 minutes means what? In the second generation. So first generation occurred in 20 minutes, second generation occurred in 40 minutes. So after 40 minutes, in the second generation, they found two types of DNA. Okay, in the first generation, they found only single type of DNA, which was hybrid or intermediate. That means in between 15 and 14. After 40 minutes, in the second generation, they found two types of DNA where 50% were light. Light means what? Light means the DNA with light radioisotope. That means 14N and 14N. And 50% were intermediate or hybrid, which means the strands are 15 and 14N. Okay, so they got two types of DNA after second generation. 50% light, that means 14 and 14N, and 50% intermediate, that means one strand 15, one strand 14. Okay, always remember the new DNA strand is synthesized from the new medium. That means new medium is 14N. Remember, the new medium is 14N, where Meselson and Stahl transferred the bacteria from the 15N medium. Yes, so after 60 minutes, so after 20 minutes the first generation, after 40 minutes the second generation, and after 60 minutes this is the third generation, where they found 25% intermediate and 75% light DNA molecule. Okay, that means gradually the number of light DNA molecule increases, whereas the intermediate or hybrid DNA molecule decreases. Very important, this slide is very important. So Meselson and Stahl observed that after first generation, after 20 minutes of transfersion of E. coli bacteria from the 15N medium to 14N medium, they found that in the new generation the DNA molecules are hybrid or intermediate. That means one strand contains 15N, one strand contains 14N, where this 14N is the new strand and 15N is the parent strand. Remember, all the new strands will be 14 and 14N. Okay, so again after 40 minutes, when they observed, they got two types of DNA, 50% light and 50% hybrid or intermediate. And after 60 minutes, that means in the third generation, they found 25% intermediate and 75% light DNA. That means you can see here gradually the number of light DNA molecule increases in every generation. Okay, so in every generation, that gradually the light DNA molecules, that means the DNA with light isotope increases, whereas the DNA level with heavy isotope decreases. Okay, now let's move to the next slide. Okay, so this is the diagrammatic representation of Meselson and Stahl experiment. So here say this is the parent DNA. So simply this is the E. coli DNA. Okay, so this E. coli DNA grew on the medium containing 15N isotope. Okay, that means this DNA contain, both strand of this DNA contain 15N isotope. Okay, so Meselson Stahl now transferred this bacteria to the medium containing 14N. Do you get my point? Here see, this is the bacterial DNA which was grown in the medium 15N. When they transfer this bacteria into a new medium containing 14N, then in the new medium, from this DNA, 2 new DNA molecule will be produced. Okay, where one strand will be old and one strand will be new. You can see clearly here, the red one is the parent DNA, that means the 15N DNA, and the blue one is the 14N DNA. If you consider the blue color is 14N and the red color is 15N, then you can see clearly in this picture that both new DNA molecule consist of one red strand, one blue strand. Blue one is the new and red one is the parent strand, and the parent means the 15N and the new strand is the 14N. Yes, this is in the first generation. Now see in the second generation. So in the second generation, 4 new DNA molecules are produced from these 2 DNA molecule. Yes, that means always from one DNA 2 DNA molecules are produced, from 2 DNA molecule 4 DNA molecules are produced. Okay, now see in the second generation, 50% are lighter and 50% are hybrid. This form is called as the hybrid. Why this is called as the hybrid? Because these DNA molecules consist of one old means one 15N strand and one 14N strand. But in the second generation, you can clearly see here that only 2 DNA molecules are hybrid and 2 DNA molecules are lighter. That means lighter means what? Lighter means both strands contain 14N DNA. Yes, but in these two DNA molecule, one strand contain 15, one strand contain 14. Again in the last one, both strand contain the 14N DNA. That means you can say 50% hybrid, this 50% lighter, and middle one, these two call as this 50% are hybrid. Okay, this way in the third generation you will get how many? Here see, you will get 25% intermediate and 75% light DNA. That means gradually the number of light DNA, these blue are light, the number of light DNA increases. Okay, so you can see here, these are the container. Okay, these are the container. So you can see after centrifugation in the cesium chloride salt, the DNA, this one is the hybrid DNA. Sorry, this one is the pure or parent DNA. Why? Because both strands contain 15N and 15N. Okay, this one is the parent DNA because both strands contain 15 and 15N. Now here see in the second container, this is the hybrid DNA where one strand is 14, one strand is 15. And in the last container, you can see the blue one. This blue one is the light DNA and this reddish one is the hybrid DNA. That means I told you that always the heavier one, the DNA with the heavy isotope found at the bottom of the container, and the DNA labeled with light isotope always found floating or remain in the upper part of the container. Okay, that means this part is the bottom part. Always you can see the heavy one is in the bottom part and the lighter one in the upper part. Okay, so in this picture this blue are the lighter DNA, and that is why you will find this blue DNA molecule in the upper part of the container, and the two middle one, these two are the hybrid. Why? Because one strand contain the heavy isotope, one strand contain the light isotope. That is why this hybrid were always found in the bottom part of the container. Okay, so this is the experiment performed by Meselson and Stahl. So there is another diagram for this experiment. So here see, this is the medium containing nitrogen 15. Yes, and this is this one. Yes, and this one is the new medium containing nitrogen 14. That means the red color is the nitrogen 15, and this orange color is nitrogen 14. Okay, so now they transfer the bacteria. This is the bacteria in the nitrogen 15. Okay, so all the DNA is initially labeled by 15N. Now they transfer this DNA to the new medium, which is the nitrogen 14. Okay, so now when they transfer the DNA from the nitrogen 15 to nitrogen 14, they observed in first generation that this is the first generation. Observe in the first generation that the products are hybrid. The products are hybrid means 50% strand contain new and 50% strand made up of nitrogen 15. Yes, here you can see clearly when they transfer the bacteria from nitrogen 15 to nitrogen 14, after first generation they observe that in the newly produced DNA molecule, one strand is the new, which is 14N, which one is the orange color, and the red color one is the 15N. In both DNA molecule, the red one is the 15 and the orange one is the 14. Okay, so after second generation they observed that in the newly produced DNA molecule, only two DNA molecules were hybrid. Only two, the first one and last one. That means only the first one and last one contain 115N, 114N. But the middle two, these two contain both strand 14 and 14N isotope. That means the middle two are called as the light DNA, and this side DNA, these two DNA molecules are called as the hybrid DNA or intermediate DNA. Okay, so now in the third generation you can see clearly here in this picture that after third generation they observed only two DNA molecules are hybrid and all the other DNA molecules are lighter DNA. That means you can see clearly here the number of light DNA molecules increases gradually. Yes, the number or amount of light DNA molecule. Light DNA means what? The DNA with light radioisotope. Okay, hybrid means what? Where one strand is 15, one strand is 14. That is called hybrid. But gradually you can see in each and every generation, gradually the number of light DNA molecule increases. Okay, so this is the experiment performed by Meselson and Stahl. Okay, so from this experiment they concluded that it is possible when the two strands of DNA duplex separate at the time of replication and act as template for the synthesis of new DNA strand having radioisotope 14N. That means from this experiment From this experiment, they concluded that when the parent DNA separate, when the parent DNA means what? Here in this experiment, the parent DNA is this one, which was labeled by 15N. Yes, during replication, when the parent DNA separate, then both separated DNA strand must serve as template strand for the synthesis of new DNA strand. Okay, the separated strands must serve as template strand for the formation of the new strand having the radioisotope 14N. Okay, here when the two strands of DNA duplex separate during replication time, each separated strand acts as template for the synthesis of new DNA strand having the radioisotope 14N, and that is why in the first generation you can see this is the parent strand where both are red. That means what? That means 15N. This DNA contain the 15N. So after first generation, in these two new DNA molecule, one strand becomes red, one strand become orange. In both DNA, one red, one orange. The orange one is the 14N and the red one is the 15N. Yes, that means they concluded that during replication, these parent DNA strands separate from each other, and both the separated parent DNA strand act as template where two new DNA strands are formed. Okay, and both two new DNA, both new DNA strands are the 14N DNA. Yes, both, here say both DNA strands are 14N. So from this parent DNA, two DNA molecule are formed where one strand 15N and one strand 14N. 14N means the new strand. Okay. So here during the second generation, 15N and 14N strands of DNA separate to function as template. Like before, during second generation, here say during second generation, these two are parent. These two are parent for the second generation. So during second generation, again this parent DNA strands separate from each other and act as template strand where two new DNA strands are formed, and that is why here you can see from this DNA two new DNA molecules are formed. Yes, each DNA molecule contain one old strand and one new strand. Here the red one is the 15N and here both orange are 14N because the parent DNA molecule contain one red, one orange, and that is why these two new DNA molecules also contain one red and one orange, and both new DNA strands are formed from the new medium, that means all from the 14N medium, and that is why all the new DNA strands are 14N 14N. Okay, so here also same, from this DNA molecule this is 14N, the red one is 15N. When they separate from each other, two new DNA molecules are formed. One became hybrid and one became lighter. Yes, this way again in the third generation, the parent DNA strands separate, and because of which two DNA molecules become hybrid and all the other product become the lighter DNA. Okay, so this way gradually the lighter or 14N DNA increases. So here say during the second generation, 15N and 14N strands of DNA separate to function as templates so that the 50% new molecules contain only 14N strand, whereas the 50% contain both 15N and 14N strand. Here in the second generation, here see in the second generation, this is the second generation. In the second generation, 50% are lighter. Lighter means 50% DNA contain only 14N DNA strand, and 50% DNA molecule contain both 15N and 14N strand. Okay, that means clearly you can say here in the second generation 50% are hybrid and 50% are lighter DNA. Okay, so in the third generation, so this way in the third generation the number of the hybrid DNA molecule decreases and the number of light DNA molecule increases. So in this way, at each replication, every time at each replication, one strand of parent DNA is conserved in the daughter while the second strand is freshly synthesized. That means in each replication, the parent DNA molecule strands separate from each other. Okay, and in that separated strand, one new strand is formed. That means you can say in each new DNA molecule, one parent DNA strand is conserved. Always the new DNA molecule retain one parent DNA strand, whereas the another strand is freshly synthesized. Okay, so from this experiment, they concluded the DNA replication process is semi-conservative or semi-conservative, where always in the newly produced DNA molecule, one strand is conserved. Which one? The parent strand is conserved and the another strand is newly synthesized. So this is our today's topic, the experiment of Meselson and Stahl. Okay, so in our next class we will discuss the mechanism of DNA replication. Till then, take care and bye bye.